we're excited about the science, but we think there's a real need to get contextualise where the science is going, share the excitement, but I think also make sure that we're on the same page. So we're going to do something a little experimental because we're talking science, and we're going to do this as a bit of a duo. We've never done this before. We've worked on all these panels before, but we thought it might just mix it up a little bit because we think we want to be a little bit more, a bit more, didactic, a bit more informal about this. So we take this obviously very seriously, but sometimes I think there can be a barrier around language, around nomenclature, and so one of the things we want to do today is to start the conversation and perhaps introduce some terminology that you might see, you might come across, and give you some more background to that might arm you to use that more powerfully, but also arm you to ask questions, because that's what we're all about. We want to hear from you and start the conversation today, but please don't hesitate to get in touch with us, and also we look forward to continuing to have opportunities to engage with the community through Holly and Emily. So I'll stop there and hand over to you.
So we'd like to share the optimism about the genetic work and gene therapy and stem cells, so hence why we're here today, and just like to reiterate the wonderful work that Holly and Emily and their families have done to bring this all together. A lot of effort goes into these conferences, so congratulations. Okay. Alright. So as I've said before, there's a lot of optimism with the gene therapies and stem cells, but if we just take a step back, and John Rudd alluded to this earlier with the natural history data, which is very important before we can get to the trials and things like that. So natural history data is where we can track the disease progression. So we're looking at visual acuity, how far one can see down the eye chart, monitoring the visual field, so the side vision measurements, and all the structure and function tests that go. We also need to look at the genetic results and correlate that with the clinical pattern to see which, in some cases, Ushers might be a bit more severe in the vision loss and others a bit more mild. So we need to have a greater understanding about that work. And there has been a few studies looking at the natural history data, particularly for the type 1 gene MY07A and the USH2As. Okay, but you're all here to learn about what's going on with the research and how we're going to potentially find a treatment or cure. And so the best treatment approach for any type of inherited retinal dystrophy, so Usher syndrome, retinitis pigmentosa, depends on the severity of the disease. So we can got a mouse with a pointer, yep, somewhere here. So it depends on where one is in the disease progression, which treatment approach would be more suitable. So early on in the disease or the condition, gene editing or potentially gene therapy might be the most suitable approach, and there would be a window period. But as the disease progresses, when the cells are a little bit more sick, then the cell replacement or the external technologies such as bionic eye might be more suitable. And a lot of this work is critically dependent on knowing the specific gene causing the type of retinal dystrophy, and John Rudd alluded to that earlier. Several of you might have seen the news articles late last... Alright, so that should be a lot better now. My apologies. Okay. So as I said, so many of you would have seen the news articles looking at the first ever gene therapy for treating blindness. And we received numerous calls about this work. Is this relevant for me? Is it relevant for my child or family members? So the gene therapy was related to a condition called Leber's congenital amaurosis, abbreviated LCA. And this is a severe retinal dystrophy causing night blindness and very poor vision in children from birth. They do get the nystagmus, so the roving eyes as well. It was first described in the late 1800s, and then one gene, the RP65, was described in 1997, and that's where it all started with the early work with the gene therapy. So it started off as an idea in the lab, progressing to the animal models, and all the way through to human clinical trials and onto the market. And Megan will touch on this a bit later about all the stages of the clinical trials to make sure that these are safe and effective before they are released onto the market. So obviously this is very exciting to have this gene therapy trial approved in America for this work. So you might ask, what is a gene therapy? And essentially it is an eye operation by a vitreoretinal surgeon to deliver a good copy of the gene, so a good copy of the RP65 gene by a specialised genetically engineered virus or a vector, or in basic lay terms, it's the delivery truck. Okay, the delivery truck brings the new DNA and is then into the eye. You're going to ask a question?
So I assume the delivery truck's been well cleaned out of the viral
Very
material.
cleaned,
Yeah.
yeah. So make sure it's going to be safe. Yeah.
So it's only got the component that needs to be replaced, and the machinery to do that?
To do that, yeah, absolutely. So this is proven to be known as safe, all in the early stages of the clinical trial, which is helpful. So essentially the RP65 trials, which were proved as a success, have paved the way for a number of retinal dystrophies. The retinitis pigmentosa, the choroidema in early clinical trials, and we can see down the bottom here that we'll be moving on to some clinical trials with Usher syndrome, which is exciting. If we get some, the next slide might be helpful so we can move on. Okay, so there are some early clinical trials for type 1 Usher syndrome related to the MY07A gene, which is really exciting, and they've been conducted in the US and in Paris. And to get to this point, there have been some challenges. John Rodell mentioned to you earlier that some genes are larger than others, and it proved that the Usher's genes were very large and couldn't fit in the specially engineered virus or the delivery truck, so they had to look at other types of delivery trucks that could bring the fresh or the good DNA. So they did find one, and now they've been able to start these new clinical trials in the early stages. With any type of clinical trial, there are strict criteria that one would need to meet, and Megan will touch on this a bit later. So for this particular trial, they were looking only at adults, so no children at this point because it was an early clinical trial, and patients needed to have the genetic diagnosis of that specific gene, because all these gene therapies and other work are all gene specific, and people have to have a certain level of vision. Okay. So all sounded wonderful and very exciting, but there was a huge price tag, and it was approximately $500,000 per eye for the treatment, and you're potentially treating 2 eyes, so it's going to be a costly little exercise and proving the next challenge for both clinicians and the health economists. Yeah. So Megan, could there be alternatives? What do you think? Could stem cells be the answer, maybe?
Yeah,
possibly.
So that's my cue. So you remember one of the first slides Lisa put up, the black background with the different stages of advance, I suppose, in the journey, and the different opportunities to intervene. And we heard about cell replacement strategies. So that's perhaps when you need to replace the cell that's dysfunctional, and perhaps other interventions like gene therapy could be tried earlier. Well, what stem cells can do is possibly not only be a source of cells for that cellular replacement strategy, but also a source of cells to work out how to deliver the gene, to address some of these technical issues that perhaps have been identified, and to do that in the laboratory. So basically to create a new way to model the disease in a dish, and to perhaps give us another tool to explore. And to do so before we start to putting cells or a particular, say, viral gene therapy into people. So what are stem cells? Thank you, Lisa. She reads my mind as well. It's great. So I just think it's really important that we do stop for a moment and talk about what these cells are, because unfortunately there is so much, at least from my perspective, misunderstanding in the community. When we talk about stem cells, I like to think about the fact that they have these kind of really cool properties. They have an ability to be able to make themselves, as well as to ultimately grow into another cell type. So when you think, next slide please, Lisa. When you think about your body, you already have stem cells in your body. In fact, in most organs in your body, there are stem cells there. And probably the one you think about or know about the most are the stem cells in your bone marrow. The stem cells, hematopoietic or blood stem cells, that give rise to red blood cells and platelets and white blood cells. Now, of course, if something goes wrong with those stem cells, you can develop leukemia, right? But we need them there functioning all the time to keep us healthy, because those other cells that they make turn over. So they only have a definitive lifespan, and they need to be keep replenished. So that's an example of a stem cell that already maintains us in a healthy state and can go wrong in disease. We do now know that there are other stem cells in our body, in our heart, in our brain, in other organs, that are there and have some kind of ability to keep the organ functioning and healthy and keep it stable, but don't have a great regenerative capacity. So they aren't able to basically replace cells like the bone marrow can, the bone marrow stem cells can. And of course a good example of that is what happens in a heart attack. Unfortunately, our cells in the heart don't grow back after a heart attack, but wouldn't it be cool if they did? So a lot of my colleagues are looking at that, how to unlock the potential of stem cells within organs. And that's often you might hear that type of stem cell biology referred to as adult stem cells. I actually prefer tissue stem cells, because I think those cells that reside in tissue usually only can make the cells within that tissue. There's another type of stem cell that you might have heard about, called a pluripotent stem cell or an embryonic stem cell. So particularly about 20 years ago, there was a lot of community discussion. Is it appropriate to use an IVF embryo, an embryo created to help someone have a baby that's no longer required? Is it appropriate for that embryo to be used in research? And this was, I don't have time today to talk about the regulations that are in place to, I think, allow that what I would argue is ethical research to occur. But this was a very exciting time for scientists, because it was the first time we could make stem cells in the dish that didn't have a restricted developmental pathway, could actually grow into anything. So we can actually coax these cells to grow into heart cells or blood cells, but importantly we can grow them into different cells of the eye. So you heard from John this morning the incredible architecture of the eye, the different types of cells within the eye. We can start mimicking that. We can even grow what is reminiscent of or representative of an early developing eye. And that's extremely powerful for research, because it means we can start modelling eye development. We can understand how the cells talk to each other. And of course we can do that with embryonic stem cells, but of course they're not from a patient. They don't really give any insight into that disease. But what if you could make a cell, take a cell from a patient with a condition, make stem cells, and then take that stem cell and make perhaps the cells that are affected in that condition? Now that was a little bit fanciful when I was doing my PhD 20 years ago. That was sort of seen as being quite an extreme goal, but we can now do that. And this is the new technology called Induced Pluripotent Stem Cells, or iPSCs, that perhaps you've heard about. It was pioneered by a gentleman called Shinya Yamanaka in Japan, and he actually won the Nobel Prize for this discovery with another colleague in 2012. And what is really incredible from our perspective about these cells is that you can take a cell from someone with Usher syndrome. You can make stem cells. You can then freeze those cells, share them with colleagues, and then grow those cells into the particular cells of the eye. And we're getting better and better at making more definitive cells from the eye, so cones and retinal cells, although to be honest, we aren't there yet in terms of completely mimicking, but we're getting better. And in a 10-year period, that's pretty good. So this is a really exciting area of science from our perspective. And I just wanted to kind of finish this section by reiterating that whilst we hear so much about how stem cells can be used for cellular therapies and cellular replacement strategies, it's really the research and the identification of potentially new therapeutics or new ways to intervene, such as, does this gene therapy idea work? So I'm going to hand it all back to you now to talk about the project. So we're working together in a collaboration. There you go, got the power.
Got the power, alright. So we can take, as Megan mentioned, we can take a little skin biopsy, and these are about 2 to 3 millimetres in size. We're not taking a big chunk of skin, it's only a very small amount, and these are generally done in the outpatient clinic.
Is that local anaesthetic?
A local anaesthetic, yep. So just small little skin biopsy. There are no stitches after the skin biopsy is being taken, just little steri-strips or little band-aids that go over the wound and a waterproof dressing, and away we go. So once it actually leaves the patient, we then give it to the scientist, all de-identified to protect the patient's privacy, and then we'll move on to the next one. And we have very, very clever cell biologists in the laboratory, and once it does leave the patients, there's no blood supply anymore. So the little skin biopsies have to be cared for like a little baby. They have to be fed, they have to be watered, we have to change their little home. If they get sick, we have to give antibiotics. Sometimes that's 3 times a day, as one of the scientists was telling me yesterday. So it is very involved with these little precious samples, and so we can only maintain some of those cell lines for a period of time, and we can only work on a certain number. But we were fortunate to receive some funding, philanthropic foundation funding, to purchase a cell culturing laboratory, or a robot, and he was given a name. His name's Pierre, and he helps the
Did Lise choose that?
I think so, yeah. From France. So the laboratory, we can see here with our gurus here, so we've got Professor Alex Hewitt and Professor Elise Peabay, who are the gurus, and we've got some stem cell biologists here working with them. And so yes, Elise did choose the name Pierre, and that helps us enormously. So we're able to then process not just a handful of samples at any one time, we can process potentially up to 100, depending on what we've got on at the time. So Megan mentioned this earlier, so it essentially can study the condition, so the Usher syndrome or a different type of retinal dystrophy in the dish, to understand what's actually going on, what's caused these cells to get sick and die, and potentially look at new drugs to fix these cells, or other genetic technologies that could prove to be a treatment for the future.
And if I could just jump in here and say that, of course, when we take the cells from the patient, we're looking at that particular condition, and Usher's is a really great example of that. But we can also use a very similar technological platform to look at other diseases, like what happens in motor neuron disease, for example. So it's not just limited to Usher's syndrome.
So we've got a range of different eye conditions. Some of the more common eye diseases, such as glaucoma, macular degeneration, so you're right, a number of different things, and there's other laboratories looking at other health conditions too. So the ability to take a skin biopsy from the arm and grow that in the lab won the Nobel Prize, so that was pretty cool. And could something else be more exciting, do you think?
Well, I think if we could really cut out the gene and replace it, that'd be pretty cool.
Yeah. So scientists are now able to do a new cut and paste technology to actually then potentially correct or prevent the disease from progressing. So we do have a nice little video. It's a BBC video, which we'll move on to the next slide to demonstrate that.
Just a day old, with a lifetime of opportunity ahead, this baby has been born into a world set to be transformed by a stunning advance in genetics, the ability to precisely manipulate our DNA. How we grow and develop is shaped by DNA, which sits in the nucleus of our cells. It's an instruction manual for how our bodies work, written in a chemical code of just 4 letters. Key sections are called genes. A spelling mistake can trigger disease. But now scientists have discovered a cheap and easy way to correct such errors by editing the code. Think of gene editing as a molecular sat-nav. It scans the DNA, searching for the error. Then it uses molecular scissors to snip through both strands, which switches off the faulty gene. Or it can repair the code by inserting a healthy copy of the gene. These techniques raise the prospect of treating, even curing, some genetic diseases. And it's not science fiction. Last month, we heard about 1-year-old Layla, whose leukemia was fixed by doctors in London who gave her gene-edited immune cells. The technology could eventually be used to treat scores of genetic conditions, like Sahana's. A faulty gene means her skin constantly blisters. It is incredibly painful and can prove fatal.
This technology holds the unimaginable dream of a cure. We really do have a hope that we can specifically correct Sahana's cells and give her a chance at a life.
The Breakthrough Prize is awarded to Emmanuel Charpentier and Jennifer Doudna.
The scientists who invented a cheap and rapid new gene editing system just 3 years ago have already been showered with awards, and labs across the world are using their technology. So what is the potential of gene editing?
And in the future, we hope that this will be a technology that can actually be used not only to understand disease, but also to cure it. We can actually change the DNA. So not only understand the information in the cell, but actually do something about it. If we see a mutation that causes disease, we have now the potential technology to fix it.
That could help patients with a whole range of genetic conditions. Their faulty cells could be removed, treated in the lab, and then healthy versions reinfused in the body. But could this science go too far? If gene editing was done in embryos, then any DNA changes would pass down the...
I think that's a talk for another day. Actually my area of interest, but I wanted to leave it there, but I just wanted to come back to the little girl who we saw at the end of the clip with her mum. She has a particular condition, it's called butterfly syndrome, so the surface of the skin is very, very fragile and it blisters and sloughs off. And in fact, what I wanted to share with you was that another colleague from the stem cell community has also been lauded with lots of awards. He with colleagues, did a proof of concept. They didn't use this CRISPR, this particular type of molecular scissors, but they used a different genetic approach to help a little boy called Hassan, who was in a German refugee camp, a Syrian boy. He has the same condition as a little girl, and he was in intensive care, ventilated, had been for months, was literally dying because he'd lost 80% of his body due to his condition, but then a massive skin infection. And through grafting, they took healthy parts of the remaining bits of his skin. They grew that skin in the lab, using obviously the stem cells within the skin, and then used a different type of genetic engineering to correct the mutation. He now has a full body of skin. He's back playing soccer, and when he sees my friend Michaeli, he comes up to him and says, "When are you going to do this?" And he has like 1 or 2 spots still here. I want this fixed. So I think we are entering a brave new world, and I think there is obviously some ethical considerations and things that we as a community must talk about. And we must talk about the risks associated with genetic manipulation, full stop. But I think it is certainly a very, very interesting time.
We could potentially move into designer babies and things like
Yes.
that, so
Yes.
a whole new topic discussion.
Yes, another day.
Another day. So there's different ways of applying the genetic editing tools, and different laboratories are trying different things. So there's one group that are taking skin biopsies and correcting them and then trying to implant them as a cell replacement therapy. And the work that we're doing is more using the genetic editing techniques along with the gene therapy method that we already know is safe. And so the work that we do at CIRA, we're the first to describe the gene therapy method with the CRISPR or cut and paste tools to genetically correct a retinal condition in the dish, and then also in the animal model, which was hugely exciting to be the first in the world to publish those results. So, moving on to, so these are all preclinical research studies, so they're not in humans at the moment, and so there needs to be careful regulation for this work, don't you think, Megan?
That's right, Lisa, beautiful segue. So what we thought was really important was just to go back to what is a clinical trial. So what we've been talking about predominantly is this area here, the preclinical research, often led by scientists working with clinicians or maybe even clinician researchers who are looking at some of these really interesting strategies and are testing them in the lab, they're testing them in animal studies before we move into humans. When we start to move into humans, it's obviously clinical research, and we believe we should be doing this in a really rigorous way. This is experimentation. It's not necessarily a shortcut to a treatment. There are so many unknowns, and as one of my colleagues says, the first-in-man studies often raise more questions than they provide answers. We also like to think about clinical trials being divided up into different stages. This is often presented as a linear kind of pathway, you know, stage 1, 2, 3, 4. But I like us to think about the fact that it sometimes has to loop back, has to go back to the lab to be improved, and then advanced again. So this takes time. It takes a lot of time. It takes a lot of money. But it's the right pathway to translate exciting discoveries and to see whether they work. The first step, though, is are they safe? So when you hear about a phase 1, phase 2 study, fantastic. Usually could be first in man, but it's usually on a handful of patients. And whilst we're hoping there's going to be benefit, what we're really wanting to see is that no one is harmed. And we want to share that information. So a well-constructed clinical trial being led by a responsible team will not only be doing this, they'll be taking their findings to conferences and sharing it, even if it hasn't worked. Because if it hasn't worked, that's equally as good, well not as good as something else working obviously, but it's equally as valuable to share, because that way other people won't be wasting their money, their time, and patients putting their health at risk potentially, or deviating from other care. So as we progress through the different phases, as you can see by the schematic, the larger the number of people we start experimenting on. Now, to come into a clinical trial, we often like to control our variables as scientists. You know, we don't want to have so many unanswered, there's already enough unanswered questions. We don't want to further compound that. So you'll hear things like inclusion and exclusion criteria. We take that really seriously, and it's important because it sets the boundaries and allows us to interpret the findings and contextualise them again. So, sorry to labour onto that, but I think it's really important, and you'll see why it's such an important issue for me in a moment. And of course, the final stages is actually when we start to ask the really exciting questions, does it work? So clinical trials are really important, and why I think they're really important, why I'm so concerned, is that a lot of stuff in the stem cell space happens outside clinical trials. You might find a website like this, you might have had a friend tell you or have encouraged you to think about stem cells, and you hop online and do some Googling. And you might come across a website like this. This one is, I think, interestingly called Returning Hope, and it's actually the subject of a lot of research I've been doing with colleagues around the rhetoric and I think the misrepresentation of the science. So this clinic is claiming to be able to link patients with experts in Southeast Asia, stem cell clinicians and experts in Southeast Asia, and they use very compelling terminology and then list that they can treat anything. So it's kind of A to Z with a bit of crazy refiling. Anti-aging, diabetes up the top, hair restoration. What's missing from the list, which is what I usually see from these clinics, is also erectile dysfunction, autism, brain injury. When it gets to eye disease, they have a special sort of breakout box of different types of eye disease that they will claim to help. And often it's the same type of intervention for any of those conditions, which we know have really different kind of causes. So it doesn't make a lot of sense biologically that this one-stop shop kind of stem cell clinic can treat not only all of these eye diseases of different pathologies, but all of these other conditions, including hair loss, really. But unfortunately, it's really tricky for, I think, people in the community to understand what is legitimate and what is not. Some of these clinic websites are so glossy, so much better than mine, and you know, appear to have more credibility than I think they deserve. Because they don't do clinical trials, they don't have evidence, they don't have peer-reviewed publications, they don't go to conferences and share their results, but what they do often do is capture patient stories on videos, and that's extremely powerful. And I hope it's true. I hope the stories we hear and the success that is celebrated is enduring, but I often wonder if it's filmed at the clinic at that time, and then I often ask myself, you know, how is this young lady now, 6 months later or 3 years later? Is she still really seeing that benefit that was claimed? These treatments are also terribly, terribly expensive. They range anywhere between $10,000 to $100,000 per treatment, and if it hasn't worked the first time, you're encouraged to come again. It's a big business. 10 years ago, when I first started to be involved in this area, the issue was around clinics in far-flung countries, which I think many of us sort of thought, obviously we wanted to raise issue with them, but we thought that the fact that they were operating in the far regions of the Ukraine or in the Bahamas wasn't terribly, well, it went to their credibility. But now, as this heat map shows, the darker the colour, the larger the number of the clinics. And as you can see, the largest number of clinics is actually in America, a country where you think they have a really high standard of healthcare and also a high standard of regulation. We also have a lot of clinics in Australia, unfortunately. So it's a big business. It's confounded by a growing number of clinics claiming to use the patient's own cells, so taking cells from you, so that therefore they're natural and maybe safer, and then using them again. And I just wanted to share one example of a case study. Unfortunately, 3 women in America had macular degeneration. Their vision was deteriorating, but they still had their license. They found out about a clinic in Florida who was called the something stem cell clinic, and one of the women actually went to a clinical trials registry and thought she was signing up for a clinical trial. They went along to the clinic. They had fat taken from their stomachs through liposuction. The fat was prepared in some way, injected into both eyes on the same day, and detached the retina and caused other problems with the eye. All 3 women are now legally blind. Now, there are a particular type of cell in fat that's interesting, and there are some really interesting clinical trials underway treating macular degeneration using cells made from embryonic or iPSCs, those pluripotent stem cells. That's real, that's happening. There's still clinical trials in the early phase. They're not a proven treatment yet, but they're promising. But in this case, it's like they've cobbled together 2 different unrelated kind of scientific observations and went, right, so therefore we will just sell this. The woman who thought she was participating in a clinical trial, she didn't realise that by asking to pay $30,000, whatever it was, to pay, she didn't think that was unreasonable. She thought that she was sort of supporting research. So I just wanted to share that story with you as a warning for how murky this area can be, and you have to do your research. And I was delighted to work with the Royal Australian College of Ophthalmologists earlier this year to produce a little flyer that perhaps provides some of that information and just gets the highlights down and starts again this conversation. So there are some tools that you can use to probe a little deeper with some marketing claims, and these kind of warnings are reiterated in the document. And I'll stop there because I know Lisa's got one more thing to say, I think.
I guess probably the take-home message is if it is part of a clinical trial, the patient should be paying for it. That's right. Probably the take-home message for it. Yeah.
That plus, I think, this scope of practice issue. If a cosmetic surgeon is claiming to help you overcome vision loss or to address motor neurone disease, think twice. These guys are, I think, conflicted. They're selling something, and we must ask for evidence.
That's right. Yeah. So it's lovely to see all of you here today, and several of you have already been involved with the research study, which has been wonderful because we learn a lot from you and motivates us in the labs and the other researchers. And the importance of advocacy about Usher syndrome and other retinal dystrophies, because money is tight, but with the advocacy of the work that you guys, Holly and Emily and other people, certainly were able to get the funding through from the government to get some stem cell money to work on these inherited retinal dystrophies, particularly Usher syndrome. So it has been wonderful, all the advocacy and support.
Yeah, and we hope we can keep this dialogue going. So we see that today is just the start, and we want to just keep you as informed as we can be. So please never hesitate to get in touch. We'd just like to finally put up our colleagues who work so closely with us. And your panel is on the...
My panel?
This big group at Sarah.
This is just a few people from the wider group, so we do have... it's a team effort, so we've got a range of people that are working in our group, ranging from clinicians, basic scientists, biostatisticians, so it is a team effort to get all these publications out, and of course the families who participate in this work. We couldn't have done it without you.
And I'm from Stem Cells Australia, which is a large, nationally funded, federally funded national stem cell initiative. But at the University of Melbourne, I work very closely with the sociologist Claire Tanner, who's in the photograph with me. I just wanted to mention Claire because I think interdisciplinary research is really important for us. That it's not just the scientists, we need to work with bioethicists and sociologists to really capture this issue and address some of these perhaps concerning practices that might stop really good research. So we're thinking kind of laterally about a lot of issues, and I think we'll stop there. Thank you.
Pediatric audiologist who deals with fitting hearing aids to newly diagnosed babies. And it used to be that we always asked them, have they had their eyes checked? Should we still be doing that? Given that you've said that there may not be any signs, is there always some sign of Usher's?
Thank you for the question. I think it is a very good idea to have your eyes checked because it's only a small proportion of the congenitally deaf population that have Usher syndrome or signs at that early age, but I guess it's more common to have other early on eye issues, such as lazy eyes or turned eyes, and we want to pick those things up as well. So eye checks occur through, in our jurisdiction, through maternal and child health nurses who will look at kids' eyes, and if there are any concerns there, they'll pass them on to be checked by an ophthalmologist, and I think that's well worthwhile having an ophthalmologist look at the eyes of a child with severe hearing impairment. We may, even in a child with Usher syndrome, think that things are completely normal.
That's the worry, isn't it?
Oh, okay. Can I interject in that? Sorry, from a parent point of view, with that, one of the other things I think that's quite common is astigmatism, if I'm correct. And from a personal point of view, Harry had astigmatism, and we put off going to the ophthalmologist for a long time because we kind of didn't want to deal with it. And he just actually really couldn't see anything quite far in the distance, which then, given the fact that he had balance issues as well as coping with the new cochlear implants, we saw a massive change in his abilities when he actually was given glasses, and we sort of were a bit like, oh, maybe we should have done that a bit earlier. So there can be some advantages in picking up some of those things to help the children.
Can I just ask, in regard to kinetic versus static perimetry, how long do you continue the kinetic as opposed to, say, Humphrey's 10-2 at a child or at an adult's level in regard to classifying an Usher's being legally blind? Is the 10-2 enough? And how long? Well, when is a kinetic test appropriate? for analysing fields?
Sure. Just for the recording, the question is regarding the type of field testing, whether we're doing a static 10-2, which refers to a 10-degree field, or a kinetic field, and also, I think I'm going to answer your question in a generic way if I can. There'll be specific things relevant, no doubt, behind the question. I think if you start using one method, the best way to compare it is to use the same method. If we change machines and we change methods, it's very hard to document change, so often we will stick with the same method. However, as I did touch on, the kinetic fields will be much wider in their range and are giving a better idea to the clinician about the person's function, particularly their far periphery. So using that to determine how well someone's spotting things off to the side and whether those areas are still working as well is the reason that I like to use kinetic perimetry. Also behind your question is what's the best test to use to gauge the level of legal blindness, which there are a few experts in the room I can see, will know that is a less than with field, as field is concerned, is less than a 10-degree central field. And that is often, I think best demonstrated with the kinetic perimetry, because the threshold perimetry with a 10-2 isn't can be very tough sometimes to perform. So I'm not answering that question very well, I know. I hope I'm giving a few tips on doing it. I think you've got to think of the reason why you're doing the field testing. And I think if you're looking for someone's function, and a kinetic field once a year or once every couple of years is a very good idea. If you're looking at the central 10 degrees, I think it's often not that sensitive to pick up change, and it looks terrible very early on in the course of the illness, and it's not as useful. Is there a follow-up question?
Okay, there is. So if you were ticking the box for the Centrelink ophthalmology report in regard to classification for legal blindness and the difference between a DSP pension and a blind pension, would you be happy with a 10-2 only? And what do other ophthalmologists consider to be adequate for that form only?
So do you want to repeat the question? Yeah. So the question again is how do I like to test fields before I tick boxes on Centrelink forms? I personally am very familiar with the Goldman fields, and so I tend to use those. I can't speak for a lot of my colleagues and what they do, but I know a lot of practices don't have Goldman fields, or they're tucked away in a cupboard somewhere and they don't get used. So they will be using the 10-2s, I'm sure. I think it will be practitioner specific on what they're familiar with.
Just on the point of the Goldman visual field, so someone can be constricted on the visual field, but they could also have these little peripheral islands out to the side, which potentially may not be picked up on the Humphrey, the 10-2, so it could be quite black on those Humphrey visual fields, but when we do the Goldman, the field of vision is actually a little bit better. They've got those peripheral islands, which functionally are very important for a patient. Yeah.
Just in regards to the genetic testing, you showed us the slide where it had sort of, with certain costs, and then it got more expensive the further you went down. I've got a few questions that really raise too, but I suppose why does it take so long? I think the last one was 6 months or something like that, and the costs. I think I know the answer to this, but who covers the cost? I'm assuming the family have to Yeah,
Okay, the question refers to the costs and the different techniques and the time it takes to investigate the genetic causes. It's costly because of laboratory time. When tests need to be set up individually for certain genes, it's really quite time consuming for the scientists involved. If it's a common test and there's lots of people participating, they'll often batch and wait till they've got half a dozen samples to test all at once, and then it's a good economy of scale. I think the last 10 years has made it, developments have made gene chip technology possible, and what we know as next generation sequencing, where computerization is very powerful at helping to unravel the gene changes. So in short, if I can explain it, they run the test many times, and then let computers reconstruct the DNA sequence that's generated by these machines. And so that's making it easier to do, but it still takes time. And I think a large amount of the time is then having the hard drive full of data and having the clinician sit down and test each of the changes that have been produced in the DNA sequence, and test it against known databases of changes that cause disease and known databases of changes that don't cause disease, but are just the variety of someone from that heritage. With the costs, the more computerised it is and the more economy of scale, the cheaper it gets. So around about $2,000 Australian, $2,500 for an exome, covers a lot of DNA. But you could get the same test for $1,000 in China, but you wouldn't get the analysis. So you've got to pay also the time for the scientists to analyse it. Who pays? The question about who pays is difficult because genetic services are on limited budgets, and they want to spend the budgets on testing people who have decisions that are made when the result comes back. So if there is a big treatment decision or a treatment option for a disease, then they're much more inclined to fund the gene testing looking for that condition. So we're talking about the future of treatments for Usher syndrome, but because they're not available on the shelf in a chemist now, it makes it harder to advocate for our patients that they should have the gene testing. However, we've been very fortunate to have access to research funds from time to time, and the Australian Retinal Disease Registry in Perth has set up some good collaborations to get some testing done on Australians or with these conditions. And Val will talk a bit later about the grants and the testing of congenital deafness and how that's been funded. And obviously when it's making big decisions about cochlear implants and just that sort of thing, I think it's easier to argue your case to get the testing. I think, Lisa's probably full of ideas with this question, is also when families are going to make future family planning decisions on the information, geneticists are much more likely to come to the party to help contribute to the cost or meet the costs.
I think Megan was going to ask.
I'll ask Lisa, I think she knows more about this than I do, but I was just thinking, I was reflecting when you were answering, John, about the whole genome project. So I can't remember exactly what year that was it year 2000? And how that, I can't remember the exact stats, but it was billions or hundreds of millions. So I think we just have to be cognizant of, as the technology, as it evolves, it gets cheaper and cheaper and more accessible. So with the conversation we're having this year may well change next year. So that
What has
my
changed over the years as well?
Yeah.
So when, so just a follow on from that, so when a family comes and they've got, concerns, they're concerned about their child, I don't know who they would see, what practitioner they'd be seeing, what clinician they'd be seeing, but who, I suppose, then will say, we advise you to have this testing, this genetic testing, I'm sure that comes from the geneticist,
That's right. So the clinical geneticists will meet patients in their clinics, which are often public hospital based, and a referral can be made from another clinician. So the person making the early diagnosis of a hearing impairment or a vision impairment can refer to a geneticist. I can only speak, my greatest knowledge is about the Victorian and Tasmanian systems, and we have some regional clinics as well. There's often a wait to meet the geneticist, and then there's considerable discussion to be had about how the testing could be used, what the testing could show, whether it's going to show a result, chance of finding a result, or having an uninformative test. And so I pass to the geneticist often to have that discussion. I know a little bit more about it than many ophthalmologists, but there's a long discussion to be had.
I think we've got time for 2 more questions.
Hi, I've got a question. I think it's probably directed to Lisa or Megan around the research. So I'm a parent of Usher type 1. I've got a daughter who's 4 years old, and she's got Usher type 1B. From a research point of view, we want to know what's the pipeline and how are the clinical trials looking in Australia and across the world as well. Is there anything in the line of hope that we could see, 5 years or 7 years or 10 years down the line? Is there anything that is going to be more promising and practical? I know we've heard of a lot of research that is undergoing, and it might take lots of, a couple of decades to become a practical approach. But I was more interested in knowing more about the RPE65 and how practical is it to become more affordable to everybody, in the near future.
Thank you for your question. So there was 2 parts to the question. So I'll just touch on the first part was the pipeline for a clinical trial specific for your daughter. I guess it's important to particularly be part of the Inherited Retinal Disease Register. Let them know that if you have a specific genetic results, often the people who are arranging these clinical trials, they want to know how many people within Australia have the specific type of Usher syndrome with that specific gene. So then you could be potentially contacted as if you'd like to be part of a clinical trial. Also keeping up with your regular eye appointments, checking for the macular edema and the cataracts, letting your clinician know. And so we know where to find you if there is a specific trial for you or other people as well. And with regard to your second part of the question was the RP65 question?
Yeah, that's right. So I wanted to know how practical is it? Is it going to be more accessible or becoming more affordable in the near future, or any equivalent to RP65?
So that certainly paves the way for other clinical trials for other retinal dystrophies. So that work from sort of bench to back onto the market was about 10 to 15 years. So they obviously had to prove that it was safe and it worked. And so there are other clinical trials studying Oxford for choronemia, phase 1, phase 2 clinical trials, and also the Usher syndrome. And given that we've established the protocols to know that the gene therapy is safe, then hopefully that might sort of take off a little bit of time to potentially for Ushers and other dystrophies. Did you want to say something?
I'm just going to make just one comment, just say it's a similar comment to the technology screening comment, is that obviously these front runners take the longest. So once we put and initiate trials using a sort of a common strategy, the ones that follow are likely to not take as long in the developmental pipeline. So I know that doesn't perhaps give you the answer you exactly wanted, but I think that's how the framework works. One of the barriers, we talk about, we cut the video around the embryo editing gene aspect, and this issue around community perception and willingness to participate, advocacy is really important. We need to have people who are saying, well, we'd like to support this area, and perhaps embryo editing, that's a step too far, but this is where it could make a difference. And if we don't have that discourse, I worry that, let's say, conservative politicians won't be very enthusiastic about supporting this kind of research. So I think it's really great for you to speak out and support if you're willing to.
Yeah, excuse me, Matt Hunter, I'm a clinical geneticist at Monash. John, I think you did a great job. I just wanted to say, just regards budgets, each genetic service has its own testing budget, and they can use that in a discretionary way. And yes, you're absolutely right, there are criteria which you've outlined, which we use to highlight who's going to get the most benefit and therefore should access that funding. But also to say that the government is really getting behind this, and the Victorian government in particular is providing a lot more funding for genetic testing. So I think it's changing very rapidly, and certainly those kind of previous barriers are coming down quite quickly.
Thank you so much. Thank you, all three of you, for answering questions. Thank you to all of you for having those questions. I'm sure there are still lots of unanswered questions. I think that they will be around in morning tea, which we're going to head off to now, which is just where the long table is behind this room.